Now, at the risk of this starting to look like the old rural scientists UNE session, it's my pleasure to welcome another UNE rural scientist and who I crossed over with at uni, David Johnston. David actually put his whole career into AGBU, working in developing genetic evaluation systems. He's been strongly focused on beef rather than sheep. But David's also at the moment leading a major project around developing genomic predictions for improving reproduction in the northern cattle industry, in the Bos Indicus and Bos Indicus derived breeds. And as an industry challenge, Repro is tough in the north, and really quite fitting, I think, for David to bring an out of a different species view to how they're addressing that problem. So, David, welcome.
Thank you, Richard. Can everyone hear me? Good morning. I'm the ring-in for the day, but thanks for Sheep Genetics giving me this opportunity. So if anyone's not interested in beef, you can grab a quick half-hour nano nap. However, while you're doing it, think what traits are important for reproduction in sheep, how can you measure them, who's going to do the recording, in what class of animals, which breeds and environments might you measure them in? How would you incorporate them into genetic evaluation? What genotyping might be required? And will you need a genomic reference population? So snooze away. So I'm going to talk about beef, and for people that aren't aware, our beef production is very much in two halves. In the north, we have our tropical breeds. In the south, we have our temperate breeds or our British breeds. Extensive production systems, very clear wet and dry, very challenging environment, hot in the south, more improved pastures and feedlots. And I'm going to talk about that northern production system and completely ignore the south. And when we're talking breeding, we're talking about variation. And it's the raw material that all of our breeders in this room are using, sheep, beef. And in beef, we've got lots of variation as well. We've got hairy ones with big horns, milk, humps, and even ones that do this for muscling. And that's genetics. That's genetic variation between breeds, but we also have lots of variation within a breed. So for something like Brahman cattle, we've got growth, carcass, meat quality, temperament, structure. But what about reproduction? It's all nutrition, isn't it? That's what we thought, that everything about reproduction was nutrition. However, we now know, and also reproduction. So particularly in our beef CRC back in 2000s, we did a lot of recording. It's a number one profit driver in Northern Australia, and it has a genetic basis, and there's lots of variation. So immediately as an animal breeder, you say, we can do something about this in our breeding programs. And this is just a very nice example from our beef CRC, where we've got two groups of cows. Now you've got tall ones, short ones, fat ones, skinny ones, but there's one big difference between those two groups of cows. We measured lifetime reproductive performance, and who can tell me which group of cows are the most fertile? Well, these cows, they skipped every second year and had 3 calves out of 6, and these cows were 6 out of 6. And there's nothing you can tell about those cows that would have told you that that was the case, except for one thing. And what's that? Record reproduction. If you want to improve reproduction, record reproduction. But how do you do that? Well, we were lucky that we discovered that we could get some early in life measures. Heifer age at puberty was one of the key traits that we could measure early in life. And unlike sheep, our cows have to get back in calf whilst lactating. So we have this issue of lactation anestrus, where the cow shuts down cycling whilst lactating, and how long she takes to start rebreeding is critical, particularly in our tropical genotypes. So again, sheep's going to be different to beef, but you have to start thinking what are the key traits in sheep that might be driving differences in reproduction. And what we found about these two traits was they were moderately highly heritable. Simply whether a cow gets in calf or not, it might only be 10% or 8% or 5% heritable. But these traits, as you'll see, they're more like 40 and 50% heritable. So we can measure these, they're related to lifetime, we select on both these to improve lifetime reproduction. So what are your indicator traits of reproduction or your component traits that you might be able to select on? The other thing is we had innovations in recording. We used live animal ultrasound of the ovaries. We looked for this structure on the ovary called the corpus luteum that shows that this cow has ovulated, and that structure then is very visible via ultrasound. When we first see that in a heifer, we use that as an indication of age at puberty. When we see it in a lactating cow, it's the resumption of her cycling. So very powerful. We've got thousands of records out of the beef CRC, and I'll talk about this Repronomics project. And also our scanners have done more than 500,000 of these scans. So they're very experienced, and we have large data sets that we're able to use. We then turn those into predictions, just like your ASBVs. We have EBVs, pedigrees, performance records, predicting differences. The more data, the better the prediction. And we're now seeing that genomics can increase those accuracies. Why? We want to buy better bulls for our commercial breeders and our seed stock breeders able to make genetic progress. This genomics, where did it come from? Mapping the human genome in 2003. We then started getting these SNP chips. We got a bovine chip very early on, albeit only in the last 10 years. And the dairy industry started using it immediately. Beef, we took a bit longer. 2017, we introduced single step that Swanee was talking about last night in sheep, and certainly we're moving forward. In terms of the dairy industry, they were able to use this genomic selection, and here's their genetic trend. There's 2008, and you can see what's happened since then. Their genetic progress has just taken off. And their genomic data, they've genotyped 1.5 million cows, and their genomic data is worth something like 25 to 140 daughter equivalents, as if those daughters were recording simply from pulling a tail hair. And that's the power. And that's when we talk about accuracy increase, it's actually equivalent number of progeny that you're getting for those traits. So they were able to increase their selection intensity and greatly reduce their generation interval. We've used it in beef, so genomics, we can come up with polled tests. We're also using it for single conditions and very powerfully in parentage assignment, particularly under multi-sire or to save on mothering up. We can use DNA now to do breed composition. So here's a drought master bull. We can use his DNA to actually determine what breed percentage he is. But making it work for selection is what we really want to see. We've got to have lots of records, we've got to do genotyping, and we've got to have advanced genetic evaluations. And the real power in beef is using it to get hard to measure traits. If we can already record it, the trouble about the dairy industry, you can't measure milk production on a bull. But if we can already measure it in beef and sheep, then we don't need genomic selection. It's these traits when we're making a selection decision where you are unable to measure that animal. And particularly in beef, it's that female reproduction. So we need phenotypes and DNA to build our genomic reference populations for these hard to measure traits. In the north, the key trait is cow weaning rate. That's our profit driver trait in our selection indexes. So that's the number one trait we need genomic selection for. However, when our breeders are selecting young bulls, we've only got very low accuracy for the female reproduction days to calving EBV. So the big question is, how do we get that accuracy up when we're making our selection decisions? And that's what spawned this project that I'll now spend the rest of the talk is how do we now fill that gap for Northern Australia with female reproduction. And the aim of the project, increase the number of animals with accurate reproduction EBVs in Northern Australia, enabling improved reproductive performance through genetic progress. And all we did was put reproduction and genomics, and I coined a word called Repronomics as an easy way of just referring to this project. How did we do it? We recorded reproduction phenotypes, intensive recording, and a precision experimental design. If you want to partition these effects, if you want to get at the genetics, often you have to make sure you've got a good experimental design, do lots of genotyping, and put it into new genetic evaluations. It's an MLA project. We've just completed it. It's been running for 5 years. a lot of collaboration between AGBU and Northern Territory and DAF in Queensland. We use 3 of the most numerically popular breeds, however their needs differ depending on whether they were previously in the beef CRC. We use research stations throughout Queensland and the Northern Territory, and we also use key seed stock herds throughout Queensland. So it was both industry and also using research stations. Just quickly, for people who aren't aware, we use Bryan Pastures, which is north of Kingaroy. We ran the 3 breeds there. We're using Brahman, Santa Gertrudis and Droughtmaster. Spyglass, which is west of Townsville, a lot further north, Brahmans and Droughtmasters there. And also the Douglas Daly, 2 hours south of Darwin. So you can see we're across several locations where we're running these research herds. We're very much using commercial production systems. Often it looks like this. However, if we get this, it can look like this. So, you know, we run these experiments across years, because certainly you can have very good years and reproduction goes up. You can have very bad years and reproduction goes down. And that's when you really start to sort the bulls out on terms of their daughter's performance. So we've got the different production systems, and clearly we have wet and dry seasons and quite challenging environments. The Northern Territory herd, it's part of a 30-year selection experiment where they've been looking at both female and male reproduction. They've been doing yearling mating, which is quite unusual in Northern Australia. And so we are interested in the genetics of this herd. They've got a large number of percentage wet rebreed rates, and their days to calving, the female reproduction EBV, they're showing a very clear genetic trend to improve that trait. So we're interested in including this herd in our research project. What bulls did we use? What we did was we went and generated the daughters on key bulls out in the industry that are currently influencing the breed. And that's important because they then become the reference population, and being related to the reference population is the way you increase your accuracy through genomic selection. So we targeted those bulls that were having high influence, and particularly if they were high influence and had very little recording, particularly of their daughters. High usage bulls and also emerging sires, bulls that were starting to hit their straps, that are starting to get large numbers. We wanted to generate 15 daughters and measure them and put that into the analysis. So we strategically sought bulls to go into this project in the different breeds. As you'll see, we also used a lot of natural mate sires, used them across years and locations so we can look at is there G by E actually happening as well for these traits. These were run as purebred, so we had cow herds of Brahmans, Droughtmasters and Santas, and they're all run together. So I'm not going to talk about it now, but that allows us the capacity to do a crossbreed, multi-breed EBVs and comparisons as well from this data. So we use lots of Brahman bulls, lots of natural mate bulls from a large number of studs. Likewise, the Droughtmasters, a lot of AI sires, a lot of natural mate bulls from a large number of studs. And Santa Gertrudis were only at the Bryan Pastures location, so fewer sires there. But again, from all the key studs, we've got their genetics represented. For example, we've used this big red Brahman bull. He's got thousands of progeny. He's like one of your rams we saw last night. So it was important we had him in the project. Here's a bull that's a US bull, so we have the capacity to do international evaluations as well. And the importance of this bull was that he's very influential, but you can see his days to calving EBV is blank. So we have no prediction of his female reproduction, so he was a key bull to include in the project. He now has 800 sons, 800 progeny, hundreds of sons, and thousands of grand progeny, and the data from this project is now seeding those analyses. What have we done? We've used intensive female recording. We naturally made our maidens and first calvers. We do that ovarian scanning. We also get weights, fats, eye muscles, hip heights, all the descriptors of body composition, and come up with age at puberty. And what we do for age at puberty is when we first see that CL in our maiden heifers, So here's some of our Brahman, Sanders, drought masters at Bryan Pastures. We record those heifers starting in February in this particular cohort, and you can see as we progressed, the percentage of heifers showing puberty is increased over the 15, 16 month period. When you turn that into age at puberty, when we first saw the CL, you can see here the distribution of Bryan Pastures in blue, Spyglass in red, and you can see a peak of heifers pubertal at about 16 months, 25 months, and some of the Spyglass heifers weren't actually pubertal to 36 months. So again, the effect of the wet season and dry season, and also the environment, the tougher environment in that north. But you can see lots of variation there, and we can actually turn it into genetic variation. So these are just least squares means for the sires, and you can see there's an 8.7 month difference in the age at puberty of the daughters of bulls under the same environment when treated alike. You will see down the bottom there, the very best bull is one of those Douglas Daly bulls from the Northern Territory selection line. And he's almost off the scale in terms of young age at puberty out of that selection line experiment. Heritability, 40 to 55%, roaringly heritable. The next trait, so we're just about getting through our how long to recycle. And this is the money trait. Puberty happens once, this happens every year. The ability of the cow to cycle, again, just an example here. This is when we start to mate, we start to record these cows, and we see that we get a peak here, but we also get this peak out here, and what's happened is we've weaned the calf. These cows were suffering complete lactation anestrus. Until we weaned the calf, we didn't break that anestrus until we weaned. And you can see then very quickly they cycle. And this is in the winter. Those cows aren't gaining condition, but we've taken that calf off. And in the north, under management, if you've got bulls out there all year round, what happens? They get in calf at the exact wrong time of year, and they'll calve in the dry season and they're going to die. The second thing is, if you retain those cows, they calve early the next time, and what do people do? They keep the heifer as a replacement. What have they now just done? They've actually selected for lactation anestrus. And so that's in the north. People have actually been inadvertently through their management possibly selecting for more and more lactation anestrus by their way they've managed those cattle. If we look at the, again, least squares means, you can see there's, again, nearly a 4-month difference in the cycling, how long they take to cycle after calving in their first calf cows, the daughters of those Brahman bulls. We see the same in the drought masters and the Sanders. And again, 25 to 45% heritable, if you can measure that trait. We also mate these cows, so we're getting precision date of birth. Obviously, you need good bull security when you're doing reproduction. We mate all the heifers, we don't cull, we don't sort, and this is often a problem. That's why we need research stations, often commercial data. Heifers are sorted and drafted, and we don't get good data for genetic evaluation and use for estimating these traits. We do a lot of recording in and out of mating, calving, branding, weaning. We measure these things for everything you can imagine. Teton udders, maternal scores, calf vigour scores, lots and lots of traits. The key reproduction traits is the days to calving, which is the EBV that we have in BREEDPLAN, but we get pregnancy, calving, weaning rates, fetal age, embryonic loss, calf losses, and lots of other traits, which I don't have time to go through. The steers, we obviously generate a lot of steers, males. They go into a MLA donor company project owned by the breed societies. They're the half sibs to the females, so we estimate those key genetic correlation between a male trait and a female trait. For every daughter a bull leaves in a herd, there's a steer as well. And so we've got both sets of traits, and it allows us to see are there antagonisms between male traits and female traits that we would have to handle. But we get lots of growth, full carcass, meat quality, and these cattle are also getting new DEXA measurements through the T's at Rocky. We've got SNP genotyping, all the project animals. We've also gone into our industry herds and EBV, genotype sires that have got high accuracy EBVs for female reproduction. So if someone's already recording it in industry, we don't need to do it again, we just need to genotype them. And that's what we've done, particularly in those good herds that are doing reproduction and also sires that have been measured in other research projects. We're using the latest SNP arrays, our high density. There's a Bos Indicus 80K, and as Swanee mentioned last night, we're using the 35K trop beef, which also has your sheep SNPs on it as well. So that's a new chip that we're using in this research. Just to show you what we've done over the 5 years, we've generated more than 5,000 calves, measured them for age at puberty, lactation and estrus, the steers, more than 350 sires from 88 studs, lots of animals genotyped, and that's now driving BREEDPLAN. And I want to spend the last little bit showing how that's working. So what we've done is BREEDPLAN, just like your sheep genetic analyses, is now single step. In fact, Brahmins was the first to go single step in 2017, primarily because of the data that came out of this project. We had to, like sheep, build the pipeline that takes a raw sample through to the analysis, rebuild the BREEDPLAN engine, add those 2 new reproduction traits for age at puberty and that lactation and estrus. We've also added a male trait for the Northern Australia because male fertility is important, so we're also using a sperm morphology trait, percent normal. That's now in the analysis as well. And the Repronomics data, both our phenotypes and genotypes, have been pivotal in this development. And what have we got? Well, how it works is we've got the BREEDPLAN engine, which is now single step. We get our reference populations where we genotype those sires, we use the genomic pedigree, and we have those research records. They form the core for these hard to measure traits. However, critical also that our industry herds, we genotype those, we get our field records for things like days to calving and our other traits, and then that forms the analysis such that we can take our young bull, he might only be 12 months old, pull a tail hair and get more accurate EBVs. And the critical is for the hard to measure traits, the traits that we can't measure on this bull when we're making those selection decisions. What does it do? Well, the first thing is, if you do recording, you increase EBV accuracies. So there's nothing magic about genomics, there's nothing magic about recording. This is a group of cows, this is their accuracy of their EBV across a whole lot of growth traits, carcass traits, days to calving, age at puberty. If you measure those traits, then you increase the accuracies. So the number one increased accuracy is do lots of recording. However, you can also use single step. So these are young bulls that don't have any recording. You can see that you get a little bit of advantage from single step and genomic selection for growth, but for something like days to calving, you can lift it from 30% up to nearly 45%, and also for carcass traits. So that's where we get the key lift, is those accuracies for those hard to measure traits. And we're getting accurate predictions on young bulls. So here's one of those bulls, and you can see for days to calving, the female reproduction EBV, here's a bull that's now no daughters, no production, with a 64% accuracy, which is really key now in the north for making those selection decisions. So we can genetically describe our young bulls so much better for our commercial producers and also our seed stock. But you can also see here there's nothing magic. If you can't record the trait, then genomic selection isn't going to help you. So we have very little reference population for retail beef yield, and even with single step, that bull's only 40% accurate. So if we want to lift this, we're going to have to build our reference population for things like retail beef yield. And change has arrived. So in the north, not a lot of people use BREEDPLAN. Probably only half of our Brahman breeders are in BREEDPLAN, so we get a lot of bulls for market without EBVs. So here's a bull at Brahman Week. They sell 1,000 bulls in 3 days. In the past, this bull had nothing. He's now got a genomic test that says he's PP, homozygous polled, and in August last year with genomics, he now gets a full set of EBVs. albeit with only modest accuracies, but we're now working hard to lift that days to calving accuracy as high as we can. So just to finalise, what have we got? The ability to better describe genetics for female reproduction. We're using the most relevant genetics that we can, driving that genomic selection. We need to keep recording in both research herds and in industry. The biggest risk is that people stop recording. Once we stop recording, our genomic accuracies will decline, unless we've got very good reference populations that will support the recording that's not happening. And for reproduction, and I think I can challenge you in sheep, there are big differences. Are you able to discover them? Certainly in this project we're able to do that, so that we can identify more profitable bulls, but most importantly that we can now start to change our breeds for improved reproduction. So just to finish, obviously this work is a large number of AGBU colleagues involved in this behind the scenes, a lot of collaboration, research station, an MLA funded project, and the Tropical Breed Societies are on board with this, and also our seed stock breeders. And just to finish up, did it make you think? So what are the important traits in sheep? How could you measure them? Who will do the recording? What animals would you record? What breeds need to be recorded? What sort of environments? How would you incorporate them in genetic evaluation? What genotyping and what reference population? So I'll leave you with that, and on that note, put it over to Kim. Thank you.
Yep. Okay, thank you, David. So for those people who thought they were going to drift off because it was about cows in Northern Australia, I trust you didn't, because there's a lot of challenging and parallel messages there, and a great bit of work that David and the team have been doing. So I've got a couple of questions popping up now, David. Has the industry engaged well in the project, given they haven't been great adopters of BREEDPLAN in Northern Australia?
Yes, surprisingly, yes. In fact, when we pick the bulls, we don't only pick people that are in BREEDPLAN. We pick bulls that are having big influence, so those that are getting used and getting lots of calves recorded. And often those people want to be in the project primarily because they trust it, and they trust the results, and they trust the integrity of the project, and in fact they embrace it because once the results come out, they know that they're very secure and very trustworthy of the data. And so we have a lot of people nominating bulls now who aren't even in BREEDPLAN who want to be part of the project.
So that in itself is a great success in terms of building recognition and acceptance of BREEDPLAN amongst those who aren't convinced it's a valuable tool.
Yeah, and of course they get as a byproduct calving ease, birth weight, weaning weight, carcass, and everything else thrown in as well.
So let's take the bottom one there, just going across breeds, or across breed groups rather, David, as to how transferable that information is into the Bos Taurus bulls.
Yep, so we've got some studies that are happening now that they're measuring age at puberty in Angus and Herefords, and again they're seeing heritabilities of the same magnitude that we're seeing here. Of course the differences aren't quite as big, but it's transferable, and there's a new New South Wales Ag MLA project that's kicking off that's going to do a similar thing that we've done in Repronomics in the south with about 5 Bos Taurus breeds. And there'll be a link in northern New South Wales that will also include the Brahman out of this project on the north coast of New South Wales to link the north with the south.
So that's another great stimulation of that project to continue to build that linkage. Getting a really technical one there at the bottom, how often do you have to scan to get that age at first corpus luteum?
We're scanning every 4 to 6 weeks, once the heifers are 240 odd kilos. So we're scanning very regularly. You can scan less regularly, but you're going to get a lower heritability trait. So you could go in and just do one scan and they'd be yes, no, are you cycling today? Well, obviously that would give you some ability to say, obviously you're going to get a big age effect, so you've got to be able to adjust for age. So if you scan more often, you're going to get a more highly heritable trait. Putting cattle through yards every 4 to 6 weeks, obviously commercial or seed stock producers aren't going to be able to do that. That's why we do it on research stations. But I'm also now trialling remote technology that is using collars, and I don't even have to muster those heifers now to be getting this data. So we're also looking, how can we do this better? And so we're looking at other devices and other technologies to get similar data that we don't have to do that regular scanning.
So things such as collars with accelerometers or motion sensors
Correct.
in them to understand animal behaviour and detect joining behaviour.
Yeah, I've had heifers
Sexual
now,
activity.
heifers that have been 200 kilos have had a collar on them now for a year and a half, and we get a recording every 20 minutes on those that will give me if they're cycling or not.
Yeah, so I wish those collars are too expensive commercially yet, but pretty exciting developments
Yep.
that all those sorts of things over time will become cheaper and more accurate. I think we might just take the last one as our last question there, David. Relationship between reaching puberty earlier and calving problems.
Yep, and that's a big issue in the north. Certainly we're not advocating that you should be selecting for shorter and shorter and shorter or younger and younger age of puberty, but equally so, we measure the weight of that heifer and her body condition. So we're not advocating low weight at puberty. We want early age at puberty, but not necessarily low weight. We don't want heifers going into puberty at 220 kilos because in the north they're going to have calving difficulties. And particularly if you don't know the birth weight AGBU of the bulls you're putting them to. So certainly we need to see that we don't look at that trait in isolation, that you also know the body weight of that heifer. But as you can see there in the north, we have, particularly in the CRC, half our heifers weren't pubertal going into mating as a 2-year-old. So, you know, you've got to look at what the issue is, but yes, you can't look at it in isolation, that it has to also be the weight of the heifer. And we don't want skinny cows cycling either, so it's not a case of tricking the genetics that we just want skinny cows, because again, in the north, those cows are at risk of dying. So we want a cow that will recycle whilst also in good body condition. That's why we do eye muscle area, P8 fat, rib fat, etc., to make sure that we understand the biology of that cow, not simply just saying we want to select for early recycling.
Yep.
Because cow survival, obviously in Northern Australia, is critical as well.
That's a really good message, I think, David, in terms of that balance and how you put traits together and keep it practical in terms of the environment you've got to operate in. And of course, those of you who have been north know that the northern environment is very, very challenging, pretty much every year, basically.
Yeah.
Could you join me in thanking David for that very stimulating discussion?